Good morning. Let's talk on carbohydrates, functions, digestion, and absorption. Carbohydrates, being the most abundant organic molecules onto the earth, they are basically defined as the aldehyde or keto derivative of polyhydro alcohols. And glyceraldehyde can be the example of the smallest carbohydrate, which is having the aldehyde and hydroxy group available. And all carbohydrates have general formula CnH2 and ON. It can also be rewritten as CnH2O whole N. It is the main source of energy, and energy production from carbohydrate will be 4 kilocalories per gram. That is equal to 16 kilojoule per grams. Storage form of energy is in the form of starch and glycogen. Then excess carbohydrate can be converted into the fat. The glycoproteins and glycolipids are the component of cell membranes and receptors. It is the structural basis of many organisms. For example, the cellulose of plant and the exoskeleton of the insects that consist of chitin. It is also a type of carbohydrate. Then glycogens are for storage. They are abundantly stored in the various body organelles. Then ribose in the nucleic acid, while the galactose in the lactose of milk. They form glycoprotein and proteoglycans also with protein, and they are present in some lipoproteins. So these are the complex association molecule of lipids and protein. Then plasma, it is also present in the plasma membrane as glycocalyx, as in sensors. And the glycophoreins is a major integral membrane glycoprotein of the human erythrocytes. So digestion and absorption of carbohydrate. Carbohydrate digestion starts right from the mouth and followed by stomach. In the stomach, there is no digestion of carbohydrates. Then small intestine under the influence of pancreatic alpha amylase. There is a complete digestion of of any of the saccharides into the monosaccharides in the form of glucose, galactose, and fructose with the help of intestinal lining, disaccharidases and the oligosaccharidases. There is a complete conversion of all type of saccharides into the monosaccharides, and now monosaccharides are ready to be absorbed via the intestinal epithelium and go into the bloodstream. So, initially in the mouth, once we take the food, the stomach, the enzyme salivary amylase begins breaking down the starch into the shorter polysaccharide, but the stay of food in the mouth is for very less time. So, enzymatic action is also for very less time. So, polysaccharides, the digesta contains still a lot of amount of polysaccharides and dextrins, sucrose, lactose, and maltose, etc. So, finally, the food reaches into the stomach. Here, there occurs the inactivation of salivary amylase. So, there is complete stoppage of action of salivary alpha amylase. Finally, the food reaches into the small intestine. The majority of starch digestion and breakdown of disaccharides occur here. The enzyme pancreatic amylase breaks down starch into the monosaccharide, disaccharide, and oligosaccharides. Then digestion of carbohydrate is completed by the enzymes attached to the brush borders of the small intestine villi. And here the disaccharides and the oligosaccharides are broken down into the monosaccharides under the influence of many disaccharidases and oligosaccharidases. So the finally the digested carbohydrate is in the form of glucose, galactose, and fructose, and via active transport now it is all set to reach into the bloodstream. Intestinal brush border contains many disaccharidases and oligosaccharidases. Lactase, maltase, and sucrases are few examples of disaccharidases. Lactose intolerance is due to the deficiency of enzyme lactase, which is responsible for hydrolysis of lactose to glucose and galactose. Lactase is present in the breast border of enterocytes. Deficiency of lactase leads to lactase intolerance, and in this condition, lactase accumulates in the gut. The irritant diarrhea and flatulence has been observed, and the condition should be recognized and treated immediately in newborns by giving the lactose-free formula diet and instead of giving the milk. And there may be the congenital and acquired causes. So as we age, the lactase enzyme will be lost. So this condition is known as alactasia or the hypolactasia. It's the most common disorder due to the deficiency of enzyme lactase. The lactase intolerance has been observed commonly in the adults, and due to the disappearance of after the childhood of most of the lactase activity in the intestinal epithelium cells. So without intestinal lactase, the lactose cannot be completely digested and absorbed in the small intestine, and it passes into the large intestine. And lactose is a good energy source for many microorganisms present in the colon. So, and they ferment it into the lactic acid and toxic products such as methane and hydrogen gas. They are produced, and they cause the abdominal cramps and diarrhea. And the problem is further complicated because the undigested lactose and its metabolites, like in the form of acetic acid, in the form of propionic acid, in the form of butyric acid. So these substances are osmotically active and increase the osmolarity of intestinal content, and which draws the water from the from the colon and resulting in diarrhea. So simplest treatment for all such condition is to avoid intake of the milk. Alternately, they can have intake of curd, because curd is rich in lactobacilli, and which is rich in the lactase enzyme. So the lactase can be ingested with the milk product. Now, absorption of carbohydrates. Carbohydrates are absorbed as monosaccharide from the intestinal lumen through the mucosal epithelial cells and into the bloodstream of the portal venous system. The two mechanisms are operated here. One is the active transport, and another one is the facilitated transport. Active transport is an uphill task, always against the concentration gradient, which requires energy. And the facilitated transport is happens as a downhill task, and which happens along the concentration gradient, so without the investment of energy. So the monosaccharides are absorbed by the intestine. The absorption rate is maximum for galactose, followed by glucose and fructose. Glucose is polar, it cannot diffuse through the lipid bilayer of the cell membrane. Hence, glucose has the transporters, transmembrane proteins along the width of the membrane. So absorption from the intestinal lumen into the intestinal cell is by the co-transportation mechanism. That is a type of secondary active transport. And there are specialized channel meant for these. These are the sodium dependent glucose transporter channels. So these are the S-glut channels. Type 1 channels are available here. And along these are type of co-transporters, and always this is the intestinal lumen. From the lumen, the glucose and monosaccharides, they come via these channels, and along with the sodium. And this mechanism is a type of secondary active transport, which indirectly requires the energy, and energy is being released from the active process of sodium potassium pump, which is also operated simultaneously, because once the glucose is there in the intestinal epithelium cells, it is being expelled out into the lumen via the sodium potassium pump, into the blood via the sodium potassium pump. And this pump the energy is being utilized for from the breakdown of ATP, and that is also supporting this co-transporter mechanism. And these GLUT channels are also present. Type 1 GLUT, as GLUT T1 channels are present in the intestinal epithelial cell, while the as GLUT 2 channels are present in the kidney tubules for reabsorption of glucose. So we'll see here the entry of glucose, how it happens right from blood into the cells, and different organelles, how they intake the glucose right from the blood. So there are two types of transporter systems are available. One is the insulin dependent transport system of glucose, and second is the insulin dependent transport system. So independent system via the GLUT 2 and GLUT 1 uh, channels, where the GLUT2 channels are operative in hepatocytes and pancreas, while GLUT1 channels are operative in erythrocytes and brain. Then insulin dependent transport mechanism through the GLUT4 channel, and muscle, adipose tissues, and heart uptake the glucose via GLUT4 channels. So have a look onto the mechanisms. So insulin receptors are available onto the cell surface. We are in the fed state, so concentration of glucose increases in the blood. So this insulin starts secreting out from the pancreas. Insulin is being uptaken by the cellular receptors present onto the various cells, and this, as soon as it binds with the receptor, it leads to certain intracellular changes and signal transduction mechanism starts up, like insulin receptor substrate 1 changes to insulin receptor substrate in the change in the conformational state, which further lead to several link reactions in between, and followed by the phosphoinositol 3 kinases activation. Once these kinases get activated, they lead to the shifting of these intracellular GLUT4 receptors to be expressed onto the surface of these cells. And now cells are set to uptake the glucose via these channels. So this is how the mechanism is operated. And GLUT4 channel uptake glucose, they are present onto the muscle and adipose and heart tissues. In the diabetes mellitus, what happens actually, there is an insulin resistance, and due to the reduction in the quantity of, and because of insulin resistance, so the GLUT channel is not able to be expressed onto the surface, so glucose uptake is not being possible in absence of insulin. So this is all due to the insulin resistance. So we have more clear account of glucose absorption in the intestinal epithelium cell. So this is the intestinal lumen. These are the intestinal epithelial cells. Then this is the blood. So from the intestinal lumen through the co-transporter, two sodiums along with the one glucose molecule, they take their entry inside the cell, and from the cell this sodium is expelled by the sodium potassium pump, while the glucose by simple facilitated diffusion method by GLUT2 channels, and this was the GLUT1 channel. And then in this picture, this is the intestinal lumen, and it's being shown here that for glucose and for fructose there are different channels. So as GLUT 1 channels are for sodium co-transporter and co-transportation with sodium, and this is the GLUT 5 channel through which the fructose takes its entry and goes into the intestinal lumen, and from the lumen via the same type of channel it diffuses out into the blood, into the portal blood. So this is the mechanism. This is the example of secondary active transport, and the sodium potassium pump is the example of primary active transport, the energy being released which is being utilized and support this secondary active transport. And these GLUT5 channels, they show the simple facilitated diffusion of fructose and mannose as well. Um, Mannose also adopts the same channels for its entry into the portal blood via the epithelial cells. So this is the glucose and other monosaccharide absorption mechanism. So after a meal, the blood glucose level immediately increases and insulin is released from the pancreas, stimulating the uptake and storage of glucose. So increased blood glucose, it stimulates the pancreas because the glucose enter via the GLUT2 channel into the pancreas, stimulates the secretion of insulin. Insulin in appropriate quantities being secreted out into the blood, and from the blood insulin is being taken up by various cells through the insulin receptor, and that facilitate the entry of glucose, and within the cell the glucose is converted into the glycogen, like in muscles and in liver it is stored in the form of glycogen. Then insulin also facilitate the uptake of glucose in the adipose tissue cells. So here there are fat storing cells. They uptake the glucose for their various purposes. Then so overall purpose is to the remove the excessive glucose from the blood and to maintain the normal range of blood glucose level. So this phenomena happens once we had a meal, and in the conditions of fasting or after several hours after a meal, the blood glucose level drops and glucagon released from the pancreas stimulates both the breakdown of glycogen into the glucose and the synthesis of new glucose. So you see it here. Once the concentration of glucose in the blood is decreased, that stimulates the pancreas to release the glucagon, that is the antagonist hormone to the insulin. It is a hyperglycemic hormone, so all hyperglycemic phenomena it will stimulate. So here in the liver, it will stimulate the gluconeogenesis to promote the synthesis of glucose from the non-glucogenic substances also. Then glycogen is also simultaneously broken up and release the glucose to meet the deficiency of glucose into the blood. So the overall glucose released into the blood and concentration is maintained, and this is how the normal range of blood glucose is maintained in the blood.